Yeah, you're good. Go ahead.
All right. So I'd like to thank Dr. Sugamaran and Provost Piskolich and President Peskovich for their introductory remarks, and to open what promises to be an extremely interesting symposium. And so we're going to kick off the first talk with Dr. Ken Mitton from the OU Eye Research Institute. And he'll be speaking to us today about lessons from COVID-19 antibody testing and targeted sequencing for rare pediatric retinal. So without further ado, Dr. Mitton.
Okay. So I'm waiting for the control tab here to let me do sharing. Okay, I'm going to go and see if this works like it did yesterday. So if this is working, is everybody seeing my screen? We're good to go. Okay, and if you can hear me okay, I'll just start off then and we'll run through this. I guess it kind of maybe falls to me to mention a little bit about the COVID-19 virus, and we'll do that in the talk. I'm really a bench scientist who uses tools to process what turns out to be a big amount of data once in a while. And one thing I've learned also doing a little project I'll tell you about looking for COVID-19 antibody testing in people is I sort of came to mind that garbage in and garbage out is a problem with all kinds of data. I see that Northwestern University and other places around the world have been modeling things like vaccine rollout and how important it will be to decide how to distribute vaccine, and that very slight changes in distribution rules can have probably a really great effect on the number of deaths prevented. And so I appreciate that all of you, especially working in that subject area, will need really good data going in to do that kind of analysis and modeling. And so start with the gold and not garbage. As a scientist analyzing data from the bench, we've experienced a lot of successes. And as I've been thinking about the successes we've had and probably other people have had, we often had to solve problems of making sure we had, gold, one of our university colors, in there and not garbage going into our analysis. And I was definitely reminded about this, like I said, when I was thinking about how I can contribute to COVID-19 as a biochemist when we were all kind of shut down from normal research for a couple of months here and around the country. So fast economical tests, it turns out, for serum antibodies of COVID-19, they came out very early in 2020, and most of these tests were not really vetted that much by manufacturers, and a lot of them suffered false positives and even failure to detect when they should be detecting antibodies in someone's serum. We chose a manufacturer's lateral flow assay, a company called Epigenetech, to test if their test would work with fingertip blood. It's a company that I was familiar with from some gene expression and epigenetics research that I'll mention in this talk. And I knew they did things like vetting their reagents before they released them. And so I thought this might be a useful project, and we indeed have a human subject research study now that we're doing in the Eye Research Institute with some of our med school students. So some lessons about garbage avoidance. First of all, for the talk, we'll go through some facts about SARS-CoV-2 for anybody that might not be that familiar with the biology. We'll talk about our fingertip blood rapid test. Is that possible? I'll think yes, it is. I've learned that much so far. And other examples that required or still require for us in our own lab having really good input. One of them is mapping a gene project where we mapped RNA polymerase to find all the genes that come on in maturing photoreceptor cells. And we also now still do DNA sequencing newer work to find the molecular causes of inherited or orphan, very rare pediatric retinal diseases that blind children. So the SARS virus that we're all interested in right now, the severe acute respiratory syndrome, coronavirus 2, says it's called COVID-19, it's called coronavirus disease 2019. As you know, it came out of Wuhan, China, is where it emerged. Not all viruses emerge from China. I believe, H1N1 happened to emerge from the U.S. Many years ago. This one's killed over 1 million people, as we all know, and probably about 210,000 persons in the U.S. We chose a lateral flow test, as I said, that we're testing right now. And the virus, if you're not very familiar with some particulars about what it does, basically cells, many of them in our airway system, in our nose, lungs, cells that have a receptor here, the ACE2 receptor, is a cell receptor that's used by COVID-19 to bind and get entry into cells, and its large spike protein that we've heard about facilitates that. The coronaviruses of this B subclass, SARS and SARS-CoV-2, MERS, they have a strangely interesting, to me as a molecular biologist, they use RNA, and they transcribe and make some proteins they need right away. They make some replicases, and they basically make the other strand of RNA. They're always dealing with RNA, and they let the cells use those RNAs to translate all the proteins they need, produce thousands of themselves, and be emerged from the cell and continue the infection. This is a Cleveland Clinic diagram, a little bit close-up view. Nice three-dimensional representation, I think, of the virus structure. Besides having its RNA in its interior, it's got a lot of specialized proteins, some membrane proteins, spike glycoprotein, small envelope protein, and this protein here, the spike glycoprotein, is the interesting one. Patients that tend to do better immune response, one study in England found that patients recovering better from the ICU from this disease tend to get antibodies to the spike glycoprotein. Vaccine makers are using the spike glycoprotein as their target. And this is the protein that's really used in the lateral flow assay as well. So lateral flow assays, early ELISA automated assays, and many of these LFA assays were found to suffer problems, mostly false positive rates that were very high, and conversely on the other side, failure to detect antibody to COVID-19 when really there was some there, which would be known from an FDA-approved clinical lab system test. So for instance, in our Beaumont healthcare system here, they have a clinically approved automated test system. Some later on, as we get to the end of the summer and right now, there are some LFA tests that are working better than the first ones. FDA-approved lateral flow assays do exist. They've existed for some time for Legionnaire's disease, other infectious diseases. There's an LFA test for AIDS that's used in the EU. They're fast, they're sensitive, they don't require power or special equipment, and if you have one that's reliable, it can be deployed anywhere. So LFA tests for antibodies in patient serum, and they can be IgG or IgM class. In the case of SARS-CoV-2, they can produce garbage data, and only a sufficient evaluation of these tests can really give you a test that gives you the gold data that you want if you're, say, examining incidence or prevalence of immunity in a population. So here's how a lateral flow assay works. There's we'll show you the real assay after they've been tested in a second, but if you would imagine there's a special nitrocellulose membrane, and on this membrane are basically fixed some capture antibodies that would capture human IgG. And also in this assay is recombinantly made spike protein, which has been labeled with basically a colloidal gold, which gives it a wine red color, is waiting in one end of this assay. And the patient, if you have a blood sample and they are positive and they have antibodies shown in red here to the spike protein, these will bind to the COVID-19 that's got a label on it, and it basically flows across this dry membrane. And the capture antibody will capture the IgG in the patient's blood sample. And if some of that IgG has indeed reacted to the spike protein and captured it, you just get an accumulation of this red color, a line. It's very similar technology to how a lot of home pregnancy tests work and the other lateral flow assays for other diseases. These are tests that we've done right here at Oakland University. And this is an example. On the top is a positive patient's test, and on the bottom is a negative group patient's test. And there's a control indicator on these assays, and this is really a chicken IgG, the capture antibody, and also a color labeled reagent. And that flow band is just to show you that you have enough liquid flow across to make it a valid test. Of course, if you don't get enough flow, your negative might not be negative. So in this case, the tests have flowed well enough. The left here is a port where the blood sample has been put onto this lateral flow cassette. This is the band for IgG positivity. And as you can see, there's a double band that's trapped and would indicate that this patient has indeed antibody to the COVID-19 spike protein. And the patient up top also had a slight IgM reactivity, and a negative person basically has no bands in those positions. This top test is interesting. This person is about 4 months after the onset of their symptoms, and interestingly, still with a little IgG hanging around, IgM hanging around. Happens to be a patient that really kind of suffered symptoms for a protracted amount of time. So far in this testing, we've got at least 25 negative test results in who should be a negative patient, people that have had the clinical arm-drawn blood test. So there's no false positives with this particular test. And so we're continuing to use collect more patients in the negative group. It's harder to find positive patients. There's more negative than positive right now, and we're still in the study. But all the positive group patients that have had COVID-19 illness so far, we've been able to detect IgG from anywhere from 1 to 4 months since these patients have been ill. And we still continue this study right now with a couple of our med students, Maron and Narain, that are fourth-year medical students in our School of Medicine here. They are running their volunteer recruitment and testing, and we're going to continue this, especially I think throughout the year, because the test is becoming, is showing us that it's reliable enough that we can now really keep using it and test more people even than we were planning. And Maron will be using that as a project to get some community information on what we have right around Oakland University. So let's move on to garbage lesson number 2. It turns out Epigen tech that makes this assay, I really decided to work with that little test on the side and get into the COVID-19 research business in a tiny way because they were a company I already used for various reagents in work like this, mapping RNA polymerase II and all gene promoters to see what genes are coming on in photoreceptors as they mature. And this is another lab experience where you had to really have gold to go into your data pipeline because there's lots of software and analysis of microarrays, and the analysis will be done regardless of what you put into the pipeline. And in this case, we'll be using chromatin immunoprecipitation. And epigenetic regulation, for anybody that's not familiar with that, is really referring to the packaging of genes that are being expressed in our cells. Our genes mostly, when they're not being used in a particular cell type, they're closed, they're silent, their DNA is wound up on these nucleosomes. There may or may not be methylated DNA, and the gene's not available to basically put RNA polymerase on and transcribe it. And usually there's a pioneer transcription factor that will recruit in histone acetyltransferases, which acetylate histones and unpack and unravel this nucleosome packing and start to make the gene available. More transcription factors can get in, and basically you eventually open up the gene's promoter, and RNA polymerase II can dock, and you can transcribe that gene. And we used mapping of RNA polymerase in genes in photoreceptors to look at genes that were off when photoreceptors are immature versus what genes are on and have polymerase on them when the gene should be on, to find genes that come on just as photoreceptors mature. And that's when the chromatin immunoprecipitation method comes into play. And in this method, cells, or retina in our case, we use a little bit of formaldehyde. You cross-link polymerase, shown in gold here, wherever it is in the genome on DNA. And then the DNA is fragmented, and it has to get sonicated. You have a good antibody, and you need a really good specific antibody to grab the polymerase, which has pieces of DNA attached to it where it's resided in the genome. These are released, amplified, labeled up, and they can either be sequenced, which is a chip sequencing, or they can be hybridized to different arrays, microarrays. In this case, we used a mouse promoter tiling array. It has 5 million probes on it that are 25 base pairs long. They're kind of tiled along the genome. There's thousands of bases of coverage around every potential promoter transcription start site. And you look to see literally where the RNA polymerase is. Now, this process, a lot of people have tried to do this kind of experiment, chromatin immunoprecipitation, and when it fails for them, it fails mostly because they can't compare samples. Because if you don't fragment all the DNA and all the samples exactly the same size, exactly the same way, if you don't have a very specific antibody and all the other processing steps, if they're not done very, very uniformly, you just don't get data that's comparable or meaningful, no matter how long or you process data or what tools you use. So in this case, we were able to control enough of these factors that we could get good data. And this is an example of good old rhodopsin, the most abundantly expressed protein in the retina. It's the light detecting protein that helps us see in rod photoreceptors. And this is a mouse, and at age P2, this gene would be expected to be off, although the photoreceptors exist. They haven't matured yet. They haven't turned on the machinery they need to detect light and see. By age 25, the retina development is done in a mouse. And these probe tracks here show you that there's plenty of RNA polymerase in the mature gene, right through the gene's promoter. This is the exons of rhodopsin. And we were able to use this to find, basically throughout the genome, about 800 genes that come on, in addition to other ones that had been found using microarray technologies. This is looking at a data set. Now we have big data, but we put gold into it. We could look at data like this for all of mouse chromosome 6. These are the polymerase tracks where it's an immature retina at P2, it's mature at P25. You can ratio these channels, and everything above a certain ratio, spiking above this threshold, is a gene that basically is coming on. And basically this worked for us really well. We discovered new promoters that people didn't know about before, and so that was kind of fun to see data work out that way. But lots of people had tried this kind of experiment, and it doesn't work unless you have good quality DNA chip fragmentation processing to go into the pipeline. Last lesson that we've had from No Garbage, lesson number 3, is currently DNA sequencing that we do with children and their family members that have some very rare orphan retinal diseases. So they're orphan diseases, they're very rare. These are even more rare than retinitis pigmentosa, which would itself be called a rare retinal disease. And we don't want garbage going into this sequencing because we really have a responsibility to get it right. We don't want any base call errors. We want to know that we have a base change that is a mutation and isn't an error in sequencing. We want high fidelity. And since we're the only place that has and made and offers this particular targeted gene test panel, even though we get it to the patients for free, we have to do it right. We're pretty much obligated to do so. So to do that, again, we want to have that gold data. And so we use an Illumina, single-colored DNA sequencing, the latest version of their sequencing. We average several hundred reads to cover any base. And we use this, what's called Q30 as the quality of data we like to use in sequencing patients. Q30 basically means that there's less than 1 in 1,000 errors per base read. And so if you do a few hundred base reads and the error rate in any one is only 1 in 1,000, you basically get rid of your error problem entirely. The ones that we target are called Norrie disease, fever, and retinoschisis. These are target genes, 8 of them here. Norrie disease and fever basically are problems in the endothelial cells of the retina, and there's a failure to grow a retinal vasculature, and you end up with a neural retina that's starved of oxygen, and blindness can result. NDP is a Norrie disease protein, is an atypical Wnt protein. It binds a receptor system built up of Frizzled-4, LRP5, and T-span12. Wnt signaling activates beta-catenin. Some of you may have heard of this pathway before. So all of these genes are in our particular test panel. There's a kinesin family member, a zinc finger that is often involved in the disease of whose function we don't really know about yet. And we do another disease that's called retinoschisis, another rare disease. Retinoschisin protein is kind of an extracellular glue that holds the layers of your retina together. And kids that have problems with this protein have a very delicate retina. They might just take a step off one stair and detach their retina and rip and tear it. So to do this sequencing and to get that non-garbage gold data, we basically have a system where we have designed really over 250 PCR primer sets that are all mixed together, and you amplify just the regions of these genes you want, about 250 base pair sizes for all 8 genes at the same time. You basically get almost 100% coverage of all the genes that we want. And we activated this service just last August, and we've successfully sequenced over 90 persons. Here's the gene view of the Norrie disease protein gene, and all the thick-banded exon areas here are covered in this case by 14 designed amplicons that basically cover all these exons and a little bit into the intronic sequence. And this is done for all 8 genes. And the kind of data we get looks like this. This is a patient with fever, and any one patient will have quite a few base changes. So in column F here is the reference genome, human genome sequence, and their change is shown here, their difference in column G. And most of these variants might be in introns. Sometimes they are in coding regions, but they change a codon, but not an amino acid. Most of these are benign changes, and some of them are common in the human population. In this case, what's highlighted in yellow here is a change that introduces a stop codon in the main receptor protein for Norrin called Frizzled-4. And here in the genome browser, this cysteine amino acid number 450 is basically turned into a stop codon, which truncates the protein. This is the reason this person has fever and a disease. And this is the disease they inherited and can also pass on. So this works okay. And again, just needing to get really good quality data into the analysis at the very beginning. So I just want to leave you with that thought of go for the gold, the golden grizzly, go for the gold data, not the garbage data. And no matter what workflow you employ in your work, we should always think and remind our students and who we're mentoring that it's important to vet our input by whatever best practices we have in our particular line of work. What can we do for quality control? We need to be asking ourselves that all the time of our input data. Do you have any built-in standards that can alert you to problems with input data? And I realize for people modeling for COVID-19 going forward in the future, I think it's very topical to think about this. The pandemic currently, I realize, has revealed that we have to worry about, there are people and forces that want to hide data or change data, sometimes for political reasons. And that's scary because it can interfere with the models and the planning for things like vaccine rollouts, and we'll have to be attentive to that. And hopefully I can see that the scientific community is aware and on top of that, and that will help us prevail, I think. And so thank you for your time. These are some of the people we work with here at Oakland University, and that's my talk for today.
Okay, thanks, Dr. Mitton, for that excellent talk. And I just, it's the lesson that Dr. Mitton's talk is particularly relevant given the high-profile retractions of papers recently in the Lancet and medicine. And so we should always be mindful of, as scientists, of producing and data of the highest quality and integrity. Okay, so I think now we'll move on to the next talk given by Courtney Ambrosic of the SAS Institute. She's a senior associate scientist, and she'll be talking about medical image analytics in light of COVID-19.
So Courtney Ambrosic, you're muted right now there. I think you just unmuted. I've given the panelists, so you should be able to share your screen when you're ready.
Okay, great. Can you all hear me now?
Yes.
Okay. And can you see my screen?
Yes, we can.
Okay. All right, I'll jump right into it. All right. Hi, everyone. My name is Courtney Ambrosic, and I'm a staff scientist